let's go through blood typing. All right. Anybody know the blood types? All right. Usually, usually women know the blood type. Usually men never do, right? Now, if you've had kids, you almost always know your blood type, right? Right. So, back in the days, we can actually do blood typing kits and everything. And they allowed us to do our own blood types. Have them. Right. But nowadays we do have blood typing kits, but they're all synthetic. It's not real blood, right? You can't test your own blood types anymore because METC is kind of worried about, some kind of safety precautions and everything. Now, when we're talking about blood types, usually we say you have two main categories, right? You are A positive or A negative, right? B positive, B negative. All right, then you have your AB positive, AB negative, and then O positive and O negative.
Mine should be thing.
All right, it should be.
Oh.
Right, it should be. Right, you're talking about like the actual, Live Well, right, Aurora, or yeah, it should be on there. Right, so check. Now these are the two categories right here. And again, the big thing you want to understand about these is like that the names that you have, like type A, that means you have the A antigen. Type B, you have the B antigen. So these antigens are glycoproteins that are found on the cell surface of our red blood cells. Now, they're unique to us, but for the most part, when we're talking about here, right, you'll see other people that have this blood type, right? So it's not kind of unique to just you, right? It's not like our matching when we have to do organ transplant, right? So you'll see people with type A, other type A's, right? So what we get is we have molecules here that are found on the surface of our cells, right? Now, this is very general for antigen. It's just a combination of proteins, glycoproteins, and glycolipids that are found on the surface of our cells that allows us to figure out if your cells are part of your larger body, right? Again, this is where we might have issues if people have an autoimmune disease, where the body itself doesn't recognize those cells are part of your body and starts attacking it, right? They're used to distinguish cell from foreign matter and allowing us to have a response. So then when foreign antigens come in, it doesn't just invade us and kill us, right? When we have a response, we have agglutination reactions, right? Antigens on the surface of the red blood cells, that is a basis for blood typing, these agglutinogens. Right now, keep in mind that what I mentioned before, right? We have free-floating antibodies already. These antibodies are what we call gamma globulins secreted by our plasma cells, which are just kind of activated versions of B cells. So we have B cells, T cells, right? Some of you guys might have heard of a helper T cell before. Helper T cells are important because they help stimulate our specific immune response, right? When they die, we don't have a specific immune response that can fight off any infections, which is bad, right? So as part of the specific defense, we have B cells that matures into plasma cells and they will form antibodies to what's invading us, right? These antibodies that are free floating in us right now are produced by these plasma cells, right? And some of them will have antibodies to red blood cell types that we don't have. If we make antibodies to what we have, we would have never been here, right? We would have been we would have never passed the first couple of days of embryology, right? You would have died before that, right? Because your own body would have attacked the red blood cells, and that's not good. So, by the fact that we're here, we know that we make antibodies to what we don't have. These antibodies will then bind to proteins, glycoproteins, and what it will do is it will then tag it for destruction, all right? Or they form antibody antigen complexes that traps, all right, the red blood cells that are foreign, right? So now you can have a clot. Either ways, it's not good, right? You either have an autoimmune reaction where we can, destroy the red blood cells, that's incompatible, or we attach to it and attach to other incompatible red blood cells that I'm introduced to. And next thing you know, we have this floating mechanism, a floating clot that can then, right, become an emboli. And then it can seed anywhere and it becomes a blood clot, which is not good, right? So these antibodies bind to antigens, forming this antibody antigen complex, which can turn into an emboli, right? A moving clot, right? Agglutinins, antibodies in the plasma that brings about transfusion mismatch, right? Agglutination is a clumping that we see as an antibody attaches to the antigens, specifically here as an antibody attaches to incompatible red blood cells, right? Again, the big thing here is understanding that we don't need a first instance, right? You don't need an accident the first time, and then the second time you will have an actual response, right? First time you get the bad blood, you will have a response that is strong, that is immediate, that is immediately life-threatening. And if we don't catch it, right, in time, and we put a whole unit of blood into you, right, you'll be throwing clots everywhere, right? And most people don't survive that. Now, when we think about ABO blood typing, it's kind of crazy, all right? Look at the difference between type O, A, and B, all right? Now, some books will make it look like there's a huge difference, but if you take a look at it, it is one sugar. That's it. Even type O has this structure right here of N-acetylgalactosamine, galactose, fucose, right? The only thing that separates A and B is this, right, second sugar that sticks out right here, right? In type B is galactose. In type A, N-acetylgalactosamine, right? So it is one sugar difference. So when somebody says like, there's a huge difference between type O and A and B, no, right? The only real difference between life and death is one sugar. So if you have type O blood, you don't have any of those sugars sticking out of it, right? If you have type B, you have galactose. If you have type A, you have N-acetylgalactosamine, right? The big thing is not that you need to know, right, the actual sugar that sticks out. The big thing is understanding just how specific everything is, right? That somehow our antibodies, right, can bind to, if I'm type O, right, our antibodies can bind to type B, type A, but somehow not bind to O, all right, because it's missing that one sugar. It's just crazy if you think about it, right? So again, right, the actual proteins, the glycoproteins that stick out, right, it makes sense that there's be a difference between the different type of proteins found with the different blood types, right? What doesn't make sense is just how similar proteins are. That the difference is just one sugar. And that one sugar is enough to cause us to have an immediate reaction and cause death if we have a mismatch, right? RBC antigens are called agglutinogens, right? And carbohydrates are what allows us to have the different types of blood types, right? Antibodies called agglutinins are the actual antibodies that are antibodies to A blood, antibodies to B blood. Now, the big thing is this, right? You never make antibodies to what you have. Right? You never make antibodies to your blood type. Again, if you did, you would have never survived to our age right now, right? You would have been miscarried early on in gestation. But the fact that we're here, right? We have survived until this point. We know this is not an issue, right? So what happens? If I have type A blood, I have type A antigens, right? We're going to see, right? In a second, if I have type A, that means I have type A antigens. I'll make antibodies to what I don't have, right? That you don't have. Right. So type A person has A antigens and B antibodies, right? Type B person is going to have type B antigen and A antibodies because, right, you have antibodies to what you don't have, right? Type AB has both A and B antigens on all their red blood cells, right? That means they make no antibodies, right? Type O has neither, right? And because of that, they are going to make antibodies to A and B, right? Now, most common type, now we usually, when we see it, we have a breakdown of where we see the actual blood type. In certain populations, in America, type O is most common, even though it's recessive, right? Type B is the rarest, right? Now, in other parts of the world, it's a little bit different. So here's the actual kind of, kit that we normally see, right? If I have type A, right, I'll have antibody to B. If I have type B, right, things like that. So this is kind of a, right, and I wish I would have brought my blood typing kit now, right? Again, right, they appear 2 to 8 months after birth. Usually we say about 6 months after birth. Maximum concentration by 10 years, right? Again, you do not form antibodies to your antigen and does not require initial transfusion, right? You don't need an initial transfusion to cause this. You are already forming these antibodies, right? All the time after the age of 6 months. And by agglutination, this is what we mean, right? So what happens is you have type A antigens sticking out. If I'm type A, I'll have antibodies to B, right? So if I'm given type B, what happens, right? The antibodies will connect, right, to other red blood cells, forming this larger clot. This larger clot then moves like an, emboli, and they can deposit anywhere, the brain, the heart, the GI tract, right? Everywhere, right? That you have blood vessels, which is everywhere, right? Now, agglutination, each antibody can attach to several foreign antigens on several different red blood cells at the same time, responsible for mismatched transfusion reactions, right? It can cause lots of, right, bleeding, lots of aneurysms, right? Lots of blood clotting events, right? Eventually, tubules, and you can have acute renal failure, acute kidney failure, everything can happen, right? And if you're given it a large dose of it before we catch it, right, you will eventually either stroke out and die or have a, MI as a result of it, myocardial infarct, all right? Again, it's just showing us, type B right here, right? What happens with the agglutination right here? Basically forming a clot. All right. All right. Type O is a universal donor. Right. Why is it the universal donor? Because it lacks that last sugar. Because of it, we say it lacks the RBC antigen. Right. The good news about this is, right, you are on speed dial at the blood center. Right. Do you want to give blood? They will open the doors for you in the weekend just so you can give blood. Right. Because you're type O and everybody can use your blood, especially if you're an O negative, right? They can use your blood. Now, if you're type AB positive, right, usually they don't want your blood, right? Why? Because if you're type AB positive, you can only give it to people that are AB positive, which is pretty rare, right? Why waste that room when you don't have to, right? So we want type O because everybody can use it. And because it lacks antigen, right, everyone can use type O blood as it does not elicit an immune response. Now, we prefer to have people that are that O negative, but O positive can be given as well. Right? The donor's plasma are gonna have, yep. What's that? They can, right? But they prefer O negative, right? And we'll talk about why the positive negative are important, right? Um, Donor's plasma may have, and they will have antibodies to B and A, right? Because they don't have type A blood, they'll have antibodies to it. Because they don't have type B blood, they'll have antibodies to it. So what we do is when we take the whole blood, we need to filter the antibodies out, right? Again, that's not bad because all we have to do is just spin it, right? As we spin it, we centrifuge it, and all the packed red blood cells goes to the bottom. And that's what we want, right? We just want red blood cells. So we just remove that, right? We'll filter it to make sure there are no antibodies that string, right? And now we have just packed red blood cells that are type O negative, right? Now, that's good. Now we give these when people are in surgery and now they're starting to have bleeding. We give them packed red blood cells. All right, that is type O. Now, we also give it to people that might be an emergency. So you get into a car accident and you're starting to bleed, the paramedics don't have time to type and match you. So they'll give you O negative blood immediately. Once you're stabilized, then they'll switch you to specific type and match, right? Because remember, if we keep giving you type O negative blood, keep giving you type O negative blood, what happens to the people that actually have type O negative blood, right? They rely on only O negative blood. You can't give them anything else, right? Well, those people are kind of screwed then, right? If there's an absence of it because we keep giving it to somebody else. So what we'll do then is type and match you. Immediately give you O negative. Once we stabilize you, we type your blood, and then we'll give you blood type specific blood, right? Now, the universal recipient is AB, specifically AB positive, right? Why? People that are AB positive, you don't have antibodies to A because you have it. You don't have antibodies to B because you have it. And if you're AB positive, you have the actual positive blood type for a rhesus factor. So then you don't make antibodies to anything, right? I can literally, if you're type AB positive, I can get anybody here, right, and give you blood immediately. Now, obviously, we wouldn't do that, but right, we could if we want. Right, RH. Right, RH is a positive negative. Right, RHD is the most reactive, and a patient's considered RH positive if the D antigen are found on the red blood cell. Right, so the positive is when we talk about A positive, that's this. Right, the D agglutinogen or protein. Right. Now, these are inherited separately, meaning like you can have type A and positive or B and negative, right? They're inherently just the way that we receive it, they're independently inherited, right? You don't just have A and then you have to have negative or positive, right? It can be either one. Now, we normally don't see antibodies to D unless we have a first instance. So anti-D agglutinins, these agglutinins are antibodies. I'm just going to rewrite that as antibodies. Right? They're not normally present, right? They can form though, but they only form when you have an Rh negative person, right? Exposed to Rh positive blood, right? This is why we blood type moms so much, right? What happens normally is this. Let's say you're A negative, right? Your husband is A positive. What happens? There's a pretty good chance your baby's going to be A positive, right? The positive presence of this protein is dominant. So if you have one, your husband gives you one of the positive genes and you have negative gene, what happens is the positive gene dominates, right? The baby will have, right, positive blood, right? The negative gene is recessive. So your husband might have to have some of the negative gene. You have the negative gene. Baby inherits both negative genes. Now baby is Rh negative, all right? But the positive gene is dominant, right? Whenever we see positive gene at all, right? For Rh, you will express it. So if your husband, right, has Rh positive, A positive blood, you have A negative blood, there's a pretty good chance baby's going to have A positive blood. What happens then? Right? Well, in the old days, we didn't really know about this. And what we would see is, right, when we have babies like this, baby is Rh positive, A positive, mom's A negative, right? The first pregnancy goes incredibly well. Baby's born, never an issue, right? But with normal birth and delivery, there's a mixing of baby's blood with mom's blood. Remember, in utero, mom and baby's blood never, ever, ever makes contact with each other, right? Never. So even though mom is Rh negative and baby's Rh positive, in utero, there's no mixing of blood. Mom doesn't know there's a difference yet. It is only with birth and delivery does the blood mix, right? At that point, mom has its first initial kind of introduction to that A positive blood, right? Mom makes antibodies to the Rh factor, right? The D, right, protein. Mom makes antibodies. And if the husband and wife stay married and they have kids again, there's a good chance a second kid is going to have A positive blood. And right, and now mom has antibodies to it, right? Mom's antibodies goes into baby's bloodstream and starts destroying baby's red blood cells immediately. And what we see is, right, first birth, delivery, perfect. Every subsequent, right, pregnancy ends up in miscarriages. Every single one, right? And of course, mom and dad are utterly devastated with each one. And until we found this out, right, we didn't know what was the cause of it, right? And the cause is because of this Rh incompatibility, right? So what do we do now? What we do now is simple. Number one, we never let the blood mix, right? So we never do, right, vaginal delivery. We'll do C-section to make sure there's no mixing of baby's blood with mom's blood, right? Because there's no mixing, mom never makes antibodies, right? And to make sure we're really safe, we give them a shot of what we call RhoGAM. Rho as in RH, right? RhoGAM, right? And what it does is that whatever, baby's red blood cells does go into mom, right? We block the receptors for it. So mom doesn't know, right? We mask it by the antibodies in the RhoGAM shot. By doing that, right, even though there might be a few stray red blood cells that does get through and goes into mom's circulatory system, right? We mask it, we have antibodies that bind to it, and then mom's immune system then never recognizes it, right? So we do both each time. So with the second delivery, right? C-section, RhoGAM, right? Third delivery, C-section, RhoGAM, right? Now, if we forget 10 years later, right? She hasn't had kids 10, 11 years. She doesn't think about it. She might have even moved to a different country, right? And now they have kids again, right? Well, what happens if we don't do the shot and the, C-section with that third, fourth child, right? That's 10 years later in a different country. Well, what happens if they do vaginal delivery again? There's a good chance that the fifth child, right, might be aborted, might be miscarried. Right? We have to do it with each pregnancy to prevent subsequent pregnancies from failing. right? Now, this is not just true with Rh negative women and Rh positive blood, but it's also true with somebody that is A negative, right? Let's say I'm A negative, right? I go into, I get into a car accident and immediately they give me O negative blood. Perfect, right? I go into the hospital now, and now what happens is, right, they type in match me, right? As they type in match, what happens is they write down on a chart, I'm A positive, right? Accidentally. There's a mistake now. What happens then, right? If I'm still bleeding, they give me the A positive blood, even though I'm A negative. What happens then? I start making antibodies to that positive blood, right? As I make antibodies, that's my first instance, right? So if on my chart now, it says I'm A positive and I get into another accident, they will give me A positive blood again. At that point, I have antibodies to it and I'll have this huge reaction, right? And the person might die as a result of it, right? By the way, this did happen in Milwaukee. I believe it was like, either St. Luke's or one of the really well-known hospitals where a nurse gave someone mismatched blood. Nowadays, this is the reason why, right? Before we do any kind of shots, any kind of injections, right? You not just have one person asking you for your name and your blood type or for an injection, right? You have two of them, right? You have one nurse come in and then second nurse to verify it, right? That's why they always check who you are, your blood type, and everything else, right? Just so we don't make that mistake again, right? Here's that RhoGAM. Oops. That we talked about, right? Leukocyte functions. Leukocytes are white blood cells. So I'm just going to write that down as WBCs. So when you hear of leukocytes, leukocytes are a general name for all white blood cells. They're the least abundant of the form elements, 5 to 10,000, as we wrote up there, right? They protect against infectious microorganisms. They have very cool looking nucleus, sometimes looking like sausages. Sometimes they're really large, almost taking up the whole entirety of the cell, right? They only spend a few hours in the bloodstream before they migrate to connective tissue. In other words, yes, we don't have a lot of them in our bloodstream. Why? Because they're usually found elsewhere, right? We don't want a large population in the bloodstream. We want it in the areas of our body where it might make contact with a bacterium first, right? We want it in our lymph nodes. We want it in our GI tract, right? We want those white blood cells to fight off the infection before it gets into our bloodstream. Because if it gets into our bloodstream, we have major issues at that point, right? They retain their organelles for protein synthesis, right? Some of them have little dots called granules. These granules contain enzymes right, that helps fight off specific pathogens, right? Neutrophils. Eosinophils, basophils are our granulocytes and our all of our granulocytes, right? Now, when we're writing this, have you ever watched a TV show, right, like Grey's Anatomy, and somebody comes in, they're sick, they're like, let's do a BMP, Chem 20, right, a CBC with differentials, or they usually say a CBC with diff, right? This is our CBC right here, all right? A complete blood count is a combination of your hemoglobin hematocrit, your WBC count, and your platelet count, right? So what we normally see is, right, the hemoglobin hematocrit, we kind of mentioned already, right? Our WBC count should normally be 5 to 10,000, right? Within the 5 to 10,000, we see a breakup that we see here, right? A differential that we see. And what does a differential mean? That means how much of that 5 to 10,000 are neutrophils? How much of it are eosinophils? I'm not quite worried about basophils because they're so small anyways. How much of these are, right, lymphocytes? how much of these are? Right, monocytes or macrophages. All right, so when you hear of these kind of, CBC with differential, right, we do a differential to get a rough estimate of what is infecting us. Is what is infecting us a virus versus a bacterium? If it's a virus, well, antibiotics are going to be useless, right? Now, we're not quite as worried about it for ourselves because we have a strong immune system, but we're damn worried about it when it's our kids, all right? When our kids are sick and they're crying in the middle of the night, right? And they, have a fever, we want to make sure we can treat it with antibiotics if it's a bacterial infection. And we want to treat it quick, right? Now, we will type and culture, right? See if we can grow the bacterium. We'll take blood out so we can culture it. 99.5% of the time, there's no, all right, sepsis. So you're not going to culture anything, all right? But we do a CBC with diff, so then hopefully we can figure out, is it bacterial infection versus viral infection, right? If it's bacterial, I can give you a broad spectrum antibiotic like augmentin, like penicillin, right? That will cover a large range of bugs, right? So how do we do this? Well, normally granulocyte count, the neutrophils, let's just say 60 to 65%, 65%. Do that middle round, right? Eosinophils, about 3%. Right? Then lymphocytes are about 25 to 30%. Right, sorry. So granulocytes are the granulocytes, right, are these right up here. And then the agranulocytes, right, the lymphocytes are about 25 to 30%. Right, or we'll just write down 30, the middle ground. And the last one, monocytes, about 5%. Right? Now, what happens is this. If I think you have a bacterial infection, in a second, we're going to discuss, right, that in some of these bacterial infections, it will cause, right, our neutrophil levels to go up. So neutrophils, I'm going to write it down right now, right, are important for bacterial infections. All type of bacterial infections will cause an increase in neutrophils. So what would happen if I do a CBC and now instead of 5 to 10,000, we're at 15,000, and our neutrophil count right here, percentage, goes to 80%. Right? That higher neutrophil count is very important. So is the high WBC count. The increase in WBC means that we are mounting an immune defense because we have an infection, right? Now, the increased percentage of neutrophils means that this infection is usually going to be bacterial in nature, right? When we multiply 80% times 15,000, we get what's called an absolute neutrophil count. That gives you an estimate of the population of neutrophils now, right? It should be elevated. And that elevation is a sign that, for the most part, this is probably a bacterial infection of some form, all right? And what happens then? I can give you broad spectrum antibiotics. I can give it to your kids, and it can hopefully start to feel better within 24 to 48 hours as an antibiotic starts to do its job. All right. Now, all right, what we're going to see next, oops, that's not what I want. All right. What we're going to see next then are these guys, lymphocytes. All right. Now, with lymphocytes right here, so neutrophils, bacterial infections, eosinophils, big one is parasitic infections. Now, right, parasitic infections, what is the most common parasitic infection in the world? The one that kills more people than anything else, any virus or bacterium combined. Yep, malaria.
Yeah.
Yep, malaria, right? So we'll see increase eosinophil counts when we have malaria, right? And then we also see, right, larger amounts when we have any kind of amoeba, right? Some other kind of a parasitic infection as well. Other types of parasitic infection. Whoops, didn't want to do that. Right. So when we talk about granulocytes and we're doing a CBC with differential, if we think we have any kind of right parasitic infection from malaria, from God forbid tapeworms, right? Right. What happens then is our total white blood cell count will elevate from 5 to 10,000 to about 15, 17,500. And then our eosinophils go to about 15%, right? Zika is a virus, right? So that's what we'll see. Now, basophils can also increase in number, but their main purpose is not to fight off anything. Their purpose is in the background. They release histamine and heparin, right? Histamine release, when it is localized, is very important, right? It's helpful. This is the reason why when we get a cut, it swells up, right? You guys ever gotten like your lip cut, right? It swells up. I used to wrestle when I was younger, and I hated wearing a mouth guard because it's hard to breathe, right? And what happens is when you wrestle and you wear braces, right? Everybody, it's like a go sign right there to just hit your mouth, right? So you just kind of hit your mouth, even when they don't want to, they want to hit your mouth, right? So after each match, my lips would just be a bloody mess, right? And what happens then is it starts to swell up, right? The swelling is actually caused by the basophils dilating that area, and then blood vessels that in that area dilate, water leaks out. What happens then? Well, it swells up, and that liquid, right, that causes swelling actually does something good. It allows you to filter all the bacterium that's exposed there, that's now found in that inflamed area, and now it's going to take it into your lymphatic vessels and then into the lymph nodes. It basically flushes that area. So then you can bring more fluid through there, and the fluid then carries the bacterium to the lymph nodes, right? And now it can mount an infection or immune defense, right, in the areas where we do have a lot of white blood cells, the lymph nodes, right? So they're important in the background here, right? Increasing more blood flow leading to the site, right? Constricting the areas after the site so then you don't have the spread of infection, right? Agranulocyte, big one being lymphocytes. Now, if your white blood cell count goes to 30, 35,000, right? That's bad, right? If it elevates that high, we're no longer worried about an immune defense. We're not worried about any kind of infection. We're worried about, all right, cancer, right? Leukemia, lymphomas, right? So leukemia is when all white blood cells are affected. Lymphomas are when the lymphocytes, the stem cells that will turn into lymphocytes, are affected, right? Lymphocytes are really important for intracellular, right? Intracellular organisms. Big one being fungi and viruses, right? Now, so we see large increases in lymphocytes when we have fungal and, more importantly, viral infection, things that go inside the cell, right? Lymphocytes present antigens to activate other immune cells. They coordinate actions of other immune cells, secrete antibodies, and always is a big one. They provide immune memory. All right. Neutrophils, when you have a bacterial infection, they go to that area. They only live for like 24 hours, right? They fight it off, they die, right? There's no immune memory if you only live 24 hours, right? So, what gives us immune memory then? Other cells like lymphocytes. Lymphocytes are going to, right, mount an immune defense against a virus, right? In the process of activating those cells, what we're going to do is keep some of those cells in reserve. as memory cells. They're not there to fight off that acute reaction. They're there to fight it off, hopefully later on, if you're exposed to it again, right? Now, we have other cells that we do clone, and they become the acute, right? Cells that fight off the infection. Well, we keep some always in reserve as memory cells for later on. Right? Monocytes also increases in number in viral infections and inflammation. All right. They leave the bloodstream and transform into macrophages. This is the first instance where we have a change in name, right? When we find it in the bloodstream, we call it monocytes. When it goes to specific areas of the body, it changes names, right? One of the things you need to know for the test is, right, types of neuroglial cells, right? So once these monocytes goes into the central nervous system, they turn into what cells? Yeah, it goes in the central nervous system, it goes to immune cells, right? Those are our microglial cells, right? And when it goes to the skin, these monocytes turn into Langerhans cells, right? So they change names. When they go into the liver, they become dendritic cells, right? So they change names depending on location. They present antigens to activate other immune cells, right? APCs. These are pretty cool, but we're not going to talk too much about it. All right. Last one's platelets, small fragments of megakaryocytic cells. The cells itself are found in the bone marrow. These are large cells, and when we need more platelets, it pinches up a portion of its plasma membrane and releases it into the bloodstream. Right? Normal blood platelet count, usually we want it closer to 400,000, right? That's the normal counts. All right. The main function of platelets, produce a seal. Oops. They can clump together, seal that area of injury. All right. Now, remember, right? As we're sealing it, it's, we're forming an initial clot, right? But that clot can still move. So we want to throw a netting over it. That's what the activation of fibrin does, right? They secrete procoagulants and clotting factors that promotes blood clotting. All right. The last thing we'll talk about is just, and we're not going to put too much detail into blood clotting. There's so many different steps. It's just glad we don't have to go through it, right? Now, this is kind of cool though, right? When you have a bleed, what happens, right? 3 hemostatic mechanisms: vascular spasm, initial platelet plug formation, and then, right, we coagulate, right, and finalize and really solidify that plug, all right, and turn it into a clot so then it doesn't move, the netting. All right. Now, what do we mean by vascular spasm? Right? Have you ever gotten a cut and it starts to throb? Right? That's a vascular spasm. Right? The throbbing event is that your body's trying to spasm the blood vessels and narrow it down. Right? As it narrows it down, you have then less blood going through, less bleeding. Right? But that is a throbbing event, are these vascular spasms. Right? What the vascular spasms do, they close up that area so then you don't need to form such a huge clot because you made that area smaller. The blood vessel that causes bleeding now is smaller, right? All right. Let's take a second break.